Okay, this lecture is to introduce you to the general virus replication cycle. So remember that viruses are obligate intracellular parasites, so they have to get into a cell in order to replicate and make more virus. And so there's basically 6 steps, and you're going to be going through these 6 steps for your virus that you've chosen to work on. The first one is attachment or receptor binding, and we talked about in the last lecture that the receptors give the virus specificity, so it's allowing it at that first step to find cells that it can replicate in. Then the virus, and this is the goofy virus here, looks like a little sunshine. Virus has to penetrate, or we call this entry into the cell. So it's got to get into the cell. And then the third step is uncoding, which means the virus has to get the genome out of the capsid and into the cell so that the genetic information can be used to make more viruses. Step 4, biosynthesis, is really where the virus is replicating the viral genome and expressing viral proteins, and not necessarily in this order, as you'll see with your specific virus. So some viruses express viral proteins first and then replicate the viral genome. Some viruses have to replicate the viral genome before it's able to express viral proteins. Assembly is really the encapsulation of the genome and other proteins. Some viruses do this, some viruses don't, and then ultimately release or exit. And we'll talk about the 2 main ways that viruses leave the cell. All right, so first is attachment. And like I said, this is the first point of specificity. So there's something on the outside of the virus that must interact with the outside of the cell. And we call these cells hosts because they are where the virus is going to get in and replicate and provide all the additional enzymes and nucleotides and amino acids and all of that for the virus. So attachment to the receptor helps determine what we call tissue tropism. And remember, that means which cells the virus can specifically infect. So this is just an illustration. Some viruses have a receptor and a co-receptor. Okay. Some viruses have one specific receptor. Some viruses use something that's not that specific, like a proteoglycan, so sugar on a protein. Not super specific, but remember this. What also determines tissue tropism is the mode of delivery. Right? So influenza isn't going to infect your liver because it's delivered to your respiratory system by the mode of transmission, airborne droplets. Some viruses have multiple receptors. So you're going to be looking at your specific virus and determining what type of receptor it has, and that will also help you determine the tissue tropism. Once we have interaction, attachment between the virus and the cell, there's a couple different ways the virus can enter. So up here in A, it's showing an enveloped virus that fuses to the cell membrane. So remember that an envelope is actually a phospholipid bilayer, and so when you learned in cell biology that membranes can fuse together or pinch apart with vesicles, this is the same idea. The phospholipid bilayer around the outside of the virus capsid here is fusing, because it's phospholipid to phospholipid, with the cell membrane and releasing that capsid with the genome into the cell. Another general method is endocytosis, and most of this is receptor mediated, which means that the virus interacts with the receptor. This interaction triggers endocytosis, so the cell is taking the virus into an endosome. Okay. And then we'll talk about, and you'll learn that somehow the virus has to get out of that endosome. So there's a few different ways for uncoating to happen. This is showing endocytosis for both an enveloped and a non-enveloped or naked virus. And down here, an example of this is poliovirus, where the viral capsid interacts with the membrane and forms a pore. And in fact, if you can see here, see if I can, sometimes I can zoom in. Yeah. The interaction between the virus capsid and the membrane allows the genetic material into the cell. So, different ways for viruses to enter or get into the cell. The exception to this is are plant viruses. So remember that plant cells have an outside cell wall. So they don't have receptors on the outside of the plant virus. So plant viruses actually enter through mechanical means, which means you basically have to break a hole in the cell wall. One of the most common ways for plant viruses to be transmitted are through insects. So what's happening here is, say this is an aphid. What aphids like to do is they like to suck the phloem. Okay? And so they will break through the cell walls or multiple cells, and as they are sucking in this phloem and sometimes into the cytoplasm, they will be releasing all those black dots, which are viruses, into the plant. You can also do this physically yourself. So when I was working with plant viruses, we would take a little bit of grit, really fine grit, mix it with our virus solution, and literally rub it on the leaf. And that's enough of a mechanical break to allow the virus to get into the cell area. The other big one is that farming machinery. So plant viruses can be a huge agricultural issue. And if you have an infected crop and you use farm machinery, which is moving through, or even farm workers who are rubbing up against the plants, you can spread plant viruses that way. If you took cell biology with me or general biology with me, you know my favorite term is plasmodesmata, which are the little kind of tunnels that connect plant cells. And then you would have your, I shouldn't do it that way, your cell wall. Oops. Oh, starting with subtitles. No, that's not what I meant to do. That's cool. How do I get out of that? What the heck? Okay. All I was trying to do was make a color and say that this was the cell wall. And so once plants, once viruses get into a plant cell, then they can travel from cell to cell through the plasmodesmata, eventually go here into the phloem, and travel throughout the vascular system of the plant. We're not specifically focusing on plant viruses in this class, but you are absolutely welcome to choose one for your research paper. Okay, so we've had attachment, we've had entry, now uncoding. And uncoding really just means getting the genome into the cell. Okay, so releasing it, remember all viruses have some kind of capsid protein structure that holds that genetic information in there. So now we want to get the genetic information into the cell so that it can be transcribed, translated, replicated, whatever it needs to be. Sometimes this happens with entry, sometimes this is an extra step. Step 4 is biosynthesis. And remember, I told you that classification of viruses is based on not only the type of genetic information they have, but how they get to mRNA. So biosynthesis means viral gene expression and viral gene or genome replication. Right? And you have to have genome replication because you're going to be packaging genomes into new virions for the next round of infection. Okay? So your job is to figure out how your virus does that and to find some cool things about how your virus does biosynthesis. So here's some examples of what I think are cool things. And when I mean cool things, I mean it's different than our normal transcription, translation, DNA replication that happens in the cell that you've been learning all through your biology career, right? So for instance, influenza and other RNA viruses use a special kind of polymerase called an RNA-dependent RNA polymerase. So you've learned about RNA polymerases before. They work with transcription. So the RNA polymerases like RNA Pol2 in eukaryotic cells are called DNA-dependent RNA polymerases because they use DNA as the template and they make RNA as the product. RNA viruses don't use DNA at all. So they go from RNA to RNA. So RNA-dependent RNA polymerase means RNA is the template and RNA is the product. I think that's kind of cool. Some viruses do translation a little bit differently. And some viruses have what's called an IRES or iris. This is this stands for, internal ribosome entry site. So instead of binding to the cap and scanning, so remember our mRNAs, you have a cap, and somewhere down here you have your AUG, and the ribosome binds and scans. An internal ribosome entry site, these viral RNAs don't have a cap protein, so they actually direct the ribosome to land, it's like a landing pad, right where translation needs to start. That's kind of cool. Or if you don't think it's cool, at least it's different than how our genes are expressed. Something else that some viruses do is called a ribosomal frame shift. So remember, open reading frames are your groups of 3 that dictate the amino acids based on the mRNA. And this is an example of SARS coronavirus. Sometimes the ribosome will actually shift back one, so you can see the C is used twice, and that opens up the second open reading frame. So it's called a -1 frame shift because the ribosome, as it's translating, goes back 1 nucleotide and then makes a new group of 3. So you have, just like we talked about in genetics, frame shift mutations or reading frames, viruses, the advantage of this is you can have one RNA molecule that can make a couple different proteins by shifting the reading frame. Something else I think is cool with biosynthesis, and I'm just trying to give you guys some ideas, so when you are doing your wiki and you have to find 2 unique things about biosynthesis, you can use examples like this if your virus does it. Some RNA viruses will actually recruit and make these double membrane vesicles you can see here in green and yellow. And what's happening is an RNA virus has to shield the RNA during replication. Because for RNA to be replicated, you have to have the 2 opposite strands, right? You need a plus, you need a minus, just like DNA replication. These are eventually going to separate, but double-stranded RNA is a trigger for cell death. So if the cell detects that there's double-stranded RNA happening, even though this is temporary base pairing, but it's happening during RNA replication, the cell will signal, hey, I'm infected, either destroy me by apoptosis, destroy me by T cells or macrophages eating me up. So viruses have hijacked membranes, and they hide in here during their replication so that the double-stranded RNA isn't noticed by the cell. And we'll talk about this more when we talk about general cell responses to viruses. Okay, so we've attached, we've entered, we've uncoded, we've done biosynthesis. One thing I want to point out to you is that viruses can have very high mutation rates. So when you learned about DNA replication, we talked about proofreading, and we have DNA repair mechanisms that help our cells monitor and fix any possible DNA mutations. But viruses actually kind of want to, let's see if I can maybe it's not right. I'm just trying to zoom in. Okay, never mind. An advantage to a virus is a high mutation rate. And we'll talk about this when we talk about vaccines and antivirals, because if our body develops an immune response to a specific virus, that virus is no longer going to be able to use our cells as a host, because we have a way of attacking and destroying and getting rid of that virus. So many viruses, especially RNA viruses, have a fairly high mutation rate. So 9 times 10 to the 6th is almost almost in every one. Oh, I can't write on here. How many zeros? 1, 2, 3, 4. So for every, sorry, I'm not explaining this very well. 1 in every 1 million nucleotides, there'll be a mutation rate for poliovirus or for influenza. Measles has a lower mutation rate. So influenza, we'll talk about, we have to have a vaccine every year for it. Which one I did that one. Because it changes so rapidly. That's because it's an RNA virus. Here are some additional mutation rates, and these aren't things you need to know. I'm just pointing out, this is per cell infection, that viruses, especially RNA viruses, can mutate at a fairly rapid rate, and that makes it a challenge for us to deal with these viruses. Assembly, which means putting the virus together, getting the virion and the genetic information encapsulated. Assembly is spontaneous, and it's what we call self-assembly. So when you get a high enough concentration of all the parts, they actually just spontaneously come together and start forming these virions and packaging the RNA. And yes, it's pretty complex. There are signals on the RNA that allow for specific encapsulation, but they have found with some viruses that you can just put a whole bunch of capsid proteins into a test tube, and it will spontaneously form virus shells or capsids. This is really cool, and we'll talk about this a little bit in using viruses in biotechnology, that you can actually put things in these empty capsids or use these empty capsids as vaccines. So the take home here is that assembly is spontaneous. Then the virus has to get out. The two main ways that viruses exit the cell is by lysis or by budding. And lysis is primarily for naked viruses, and budding is for enveloped viruses. So let's take a look at these. Lysis is just what it sounds like. The cell kind of self-destructs. I like to say it kind of explodes. So here you can see the outline of a bacteria, and all of these phage particles just bursting out. That's the word I was using, or thinking of. So you can get a viral burst, just this massive outpouring of viruses. Enveloped viruses, because they have to have that phospholipid bilayer around them, will pinch off from various membranes. So these are showing you viruses pinching off from the cell membrane. This is HIV, and this is influenza. And what I want you to see is that this coat of the membrane surrounds it and becomes the virus envelope. Here's all these influenza viruses budding off of this extension of the cell membrane. Maturation is a step for some viruses. And all this means is that sometimes viruses have to go through one more step. This is an example of HIV. This is influenza. That in order to be infectious, they may need to go through one more conformational change. So maturation of HIV is the formation of the capsid. Maturation for influenza is actually a cleavage of the HA protein. And without maturation, these viral particles are not infectious. Okay, so I'm just seeing where I am. One more concept I want you to be aware of is this idea of particle to PFU ratio. PFU means plaque forming unit. So a plaque forming unit is basically an infectious virion. Because of high mutation rates, some particles, virus particles, are not infectious. So people have studied, of all the particles that a virus makes, how many are actually infectious. So you can see herpes virus has a huge range. So there's 50 to 200 particles made for one infectious virion. And that can seem quite wasteful, but it also means that if there are advantageous mutations, how do I want to say this? It didn't work to say it right. The high mutation rate can be an advantage because if there's a mutation that allows the virus to escape the immune system, then that virus is going to be able to continue to replicate. So for viruses, making a lot of particles that have slightly different genetics, which is most likely why they're not infectious, is okay, because if there's a mutation that gives it an advantage, now it can go on and infect additional hosts. Whereas a cell, mutations can be detrimental, right? So a virus might produce 10,000 particles. Maybe only 1/200th of those are infectious, but it's enough to carry on the infection. I don't know if I'm explaining that very well. But I just want you to be aware of this idea that not every particle produced by a virus infection from a virus replication cycle is infectious. And the way we determine plaque-forming units are these little dots here. Right? So remember that viruses have to grow in a cell culture or in some kind of cells if we're working with them in the lab. So say these were my cells, all nicely grown in the little circle dish. Okay, and we call this a mono layer of cells. And then, oops, let's try to. and then we add some virus to it. So say a virus infects this cell, and it infects this cell, and it infects this cell. And then what's going to happen is the virus is going to make more virus and go on to the next neighboring cells. And so what you end up with is, instead of your nice monolayer, you kind of have some, let me see how I'm going to draw this. You have some holes. Right, I'm not drawing this very well. What I'm trying to show you is, let's zoom in. Can I zoom in? Nope. Nope. How come I can do it sometimes and I can't other times? Alright, never mind. You can zoom in. All these little dots are representing a single virus. So a virus infected and moved to the neighboring cells, and then when we stain, all the live cells are stained purple, and the holes in the monolayer are caused by one infectious virus. So we can quantitate, doing a series of dilutions, how many plaque forming units that hole is called a plaque. So if you've taken microbiology, this is kind of analogous to colony forming units, right? So when you plate your bacteria, you get different colonies, or you get multiple colonies growing, and each colony represents from one bacteria. So it's kind of the same, it's just instead of making this little mound of bacterial cells, you're actually destroying some of the you're destroying the host cells that the virus has infected. Please let me know if you have questions, if that didn't quite make sense. Okay, so that concludes viral replication cycle. You've got the 6 steps that you need to look up for your specific virus, and especially the biosynthesis part. Find a couple things that are kind of cool about your virus, or something new you're learning about your virus to share with the class. All right, thank you.